Hey there guys, welcome back to Cambridge Biology Revision. If this is your first time here, then my name is Tom and I do videos on whole topics or as big a topics as I possibly can for the Cambridge Biology, and this is an A-level video. It's particularly focused on the microscopes practical section of Unit 1. There is another video which has got all of the content for that topic, but for this video I'm going to try to teach you how to use a microscope for practical paper 3 without actually having a microscope in front of you. So let's get started. So then guys, as mentioned, we are on microscopes as a topic. If we take a look at what we're going to do today, effectively the whole lesson is based around using the microscope and the skills that are associated to it. Those skills are as follows. You need to be able to use the microscope and focus it. You need to be able to calculate with magnifications either from the microscope or theoretically in the paper if you get given a picture. Evaluating the errors that quite often come up, and I'm going to make sure to show you some exam tips that you might be able to use in your exam at the end of the video. And then you also need to be able to use a micrometer and produce cell diagrams from the microscope. Again, it might be given to you as a picture or as a question, but it can also be given to you as a practical, so it is best to know how to use the microscope to do those things. If you haven't been to one of my videos before, just very quickly, bold means it's an important word, red means it's probably a new word for A-level, and if it's in italics, I'm giving you something that you need to replicate exactly, so you write it exactly the same as I have in the exam for marks. And these are, I'll disappear for a second, the exact specification points given from Cambridge for this practical. All right guys, so let's make a start. First and foremost, we need to be able to make a slide to put into our microscope. This is quite straightforward and it's mostly theoretical. I'll explain why in just a moment, but you need a glass slide. That's the main object that goes into the microscope, and it's the big white rectangle you can see at the bottom. And then on top of that, you of course put a cell material or a specimen. That's the thing you're trying to look at. On top of that, usually, but not always, you have a stain. The reason you have a stain or a type of ink or liquid is to colorize certain parts of cells or tissues that you're trying to look at. Without that, most of the time, cells are not, you can't see what's in them. And so it's a really important one to remember, and occasionally Cambridge do test on that. Last but not least, to make sure your whole specimen is mostly 2D, although it's impossible to be completely 2D, you kind of squish all of that material in between two pieces of glass by adding a cover slip on top. Now, as I mentioned, don't stress too much if you don't have access to a microscope. That's because in the Cambridge Practical Paper 3, it's very unlikely that they're going to ask you to actually prepare your own slide, because they send all of the slides to students for the practical exam. That's because they need to keep all of the slides the same, so everybody's looking at the same thing. So don't stress if you don't have access to a microscope right now. You can just kind of learn that slide preparation in theory and know that you'll get given a slide that you'll have to focus in the real exam. Let's move on to having a look at how we focus the microscope. So, how do you get that microscope focused? We're going to look at the theory, and then I'm going to show you some videos to really help you to put together that idea of what you need to do in the practical. First and foremost, you need to be able to, sound silly, but turn on the microscope. Don't forget, there is a big button underneath, usually on the side, where you flick on your microscope, and you need to orientate the slide with the slide wheels. I'll show you what I mean by orientating in just a second. After you've got the microscope turned on, the lights on, then you need to focus the microscope. Here's a really important part, and I'm going to stop for a second to make sure you understand. There are usually 3 or 4 different types of objective lens. When you look through the objective lens, that is the part of the microscope doing the magnification. You also have an eyepiece lens, but it remains the same and you can't change it, and it's a multiplication of 10. And the objective lenses usually sit at 4, 10, 40, and sometimes 100, which if you do 4, which is the objective lens, times by the eyepiece lens, which is 10, you would be magnifying your image 40 times. In almost all of the Cambridge practicals, the best objective lens to pick is the 10 times size, so the 10 times objective lens, which gives you a total magnification of 100 times. Once you've done that, you're actually focusing on the magnification of the specimen, and that involves turning the focusing wheels. I'll show you what those are in just a second, don't fret. The reason you're turning those focusing wheels is you're effectively moving your specimen up and down to make sure you're in the right frame for the magnification to take place. So how do we do that exactly? Let's take a look. I'm going to disappear in a second, but I've just pointed out for you what the orientation wheel is. This is turned to make sure the slide is in the middle. It's pretty straightforward to use. And then I've also pointed out which one is the focusing wheel. As you can see in the first video, or you're about to see, I'm turning that, and on the right hand side you can see what you would be able to see in the eyepiece as you're turning the focusing wheel and bringing the frame or the specimen closer to the objective lens where it will be magnified. You will notice in this video I swap between using the coarse focus, which is a big wheel there, and using the fine focus at the end to make sure I'm exactly the right place. And there you have it, guys. That's how you focus on the specimen. It seems really tricky when you're actually doing it, but with a little bit of practice you will get there. So don't give up and make sure you keep practicing before you get to that exam if you do have access to a microscope. If you are following along with my course, or even if you're just studying on your own, make sure that you do take a break to practice independently focusing a microscope on a microscope slide. This part isn't something that you can do theoretically, and it is really something that you're going to have to do in your Cambridge exam. If you don't have access to a microscope at home or even at school, there are a lot of science museums and science facilities that you can go to look at how to focus a microscope. Don't give up, make sure you practice, and let's move on to some more of the skills that we're going to need now. Okay, next, everybody's favorite part of biology, using maths in biology. Let's have a look at how we calculate our magnification that we've just learned how to do on a microscope. Usually this is theoretical, so you need an equation triangle for this. It's called the I-am triangle, which is the easiest way to remember it, by the way. I stands for image. Now, when we say image, it's what you're looking at through the eyepiece, or what you can see, which is usually presented as a diagram on the piece of paper or the exam that you're looking at. A, which is actual, is the size of the real specimen, and M is the number of times that you've magnified it. If you're looking for I, you cover up I, and you end up with a line across the bottom, which is A times M, or actual times magnification. And then for either of the other two, you cover it up. If you cover up the A, it's I over M, or image divided by magnification. Last but not least, if you want magnification, you cover up the M, and it's I over A, image divided by actual. Let's have a look at how we use that equation triangle in an example, and that will help you to understand exactly what it is you need to do. Now, just before I start putting cell diagrams up on the board, one of the things you're going to see when we start doing this calculation are these following units. You've got two that you probably remember from, any subject using a ruler. Centimeters are the big units that you see on a ruler, and 10 times smaller than a centimeter is a millimeter. Those are mostly common to students, but two new ones that you may not know are micrometer and nanometer. Micrometer uses a very weird symbol, which is the Latin for micro. It's like a U, but you pull down the left-hand side of the U. Make sure that you know how to write that, and I've given it to you just over here to make sure you do. Nanometer is just nm. Now, moving between those units is what often confuses students. We know from a ruler that centimeters is 10 times bigger than a millimeter. So if you move from centimeters to millimeters, you times your answer by 10. 2 centimeters would be 20 millimeters. But if you move from millimeters to micrometers, it's 1,000 times different. That means 1 millimeter is 1,000 micrometers. Be careful, if you're given centimeters, that means a centimeter is 10,000 times bigger than a micrometer. Last but not least, we have a unit which is even smaller beyond that. We have a nanometer. A nanometer is 1,000 times smaller than a micrometer, which means it's 1,000,000 times smaller than a millimeter and 10 million times smaller than a centimeter. I've given you a nice diagram up here on the left, which is what you can use and you should make a note of to move between the different units. Have a practice if you haven't seen this before. As promised, here's an example calculation of something that you would see in an exam. The magnification calculation usually gives you an image that you're going to work from. Don't worry if you don't recognize it, because they'll either put a bar across that image so you know you can measure something, or they'll tell you what it is in the exam question. Don't panic. The image shown here is of a rhizome found in a plant stem. And here's why we need to make sure we're reading slowly and carefully in all exam questions, but particularly when we're calculating. The rhizome is 12 micrometers in length. What we mean by it is 12 micrometers in length is that we have the actual size of the thing that is shown. We have the actual, and we're asked to calculate the magnification used to produce this image. What do we need to make sure we do? Use the equation triangle. We are calculating m, which means we're dividing the image size by the actual. Luckily, we already have one of those things, the actual. If we already have the actual, then that means our measurement is going to be of the image. Take out your ruler and measure across the length of the bar. It doesn't need to be super accurate. Usually Cambridge will give you 1 millimeter either side of the correct value, so a little bit of deviation. In this example, I calculated it with millimeters because it's more precise than just using big units, which are centimeters. I measured it as 60 millimeters, but here's the kicker. Millimeters cannot be divided by micrometers because it's a different unit, which is where we need to use that unit conversion that you've just made a note of in your textbooks, or I hope you made a note of in your textbooks. So 60 millimeters is the same as 60,000 micrometers. I times my answer, or my image that I calculated, by 1,000, which is how we move between millimeters and micrometers. Finally, I can actually carry out that division, which gives me an answer of 5,000 times. Just another note, in answers, you're either given the unit that you need, or you have to put your unit that you've worked out. Don't forget to write down the symbols or unit standards that we just learned in the previous slide if you don't get given the unit in the answer bar. In this case, it's magnification. There is no unit, so our answer was 5,000. Now, if you are following along with my worksheets and my course, now would be a good opportunity for you to stop and carry out the calculations from cell images using the magnification equation. If you don't have my worksheet, don't worry, just go and find some past exam papers on the internet and have a go. All of this will be found in paper 3 for Cambridge. Now, both of the things that we've looked at so far, which was focusing a microscope with slide preparation, as well as using the magnification equation triangle, were things that you should know from GCSE. What we're about to look at, which is called a slide micrometer, is something that is new to the A-level, or you will not have seen this in an IGCSE paper. A micrometer, which sounds like the unit of measurement but is actually a piece of equipment, is a glass slide that has a single 1 millimeter unit on it, with lots of divisions in between. We use this to calibrate a microscope to work out the size of an actual specimen without using the magnification of triangle. So what we're doing is using the microscope to calculate its real size rather than using the magnification equation triangle on a piece of paper. Now, if I zoom in for you, just so you're aware of what it is you're looking at on that micrometer slide, you're looking at a circle, which is usually found in the middle, and on there you have a unit of measurement. It's a standard, something you can work from. The whole unit is 1 millimeter in length, and then every subdivision you would divide. Here we have subdivisions and there's 10, and so each one of those is 0.1 millimeters. Often there are even smaller subdivisions, which would be 10 within each 1/10, which would give you 0.01 millimeters. Have a look at a micrometer if you get an opportunity through the microscope. Make sure you understand exactly what we mean here, because we need to know before we move on to the next stage, which is using this to calculate the actual size of a specimen. Now, the best way to show you how to use a micrometer is to show you 2 videos. I'm going to play the first video now and then explain exactly what's happened, then another video, and if you're unsure afterwards, I've written out the instructions for you so you can read them carefully. So, the first video, let's watch. So the first thing I'm doing is I'm lining up my graticule, which is what's the small line and is found in the middle of the eyepiece, with the micrometer, which are those bigger units that you can see in the background that I focused on first. Once they're lined up, I can take a measurement of the micrometer against the graticule. I'm going to stop the videos and show you an example of that very quickly. So what I've done is stuck up a cartoon of a slightly different picture and scaled micrometer and graticule. In this example, the graticule is the large crosshairs in the middle, and the micrometer is the smaller bar which is lined up against the left-hand side. When we're calibrating, we can see that once lined up, the micrometer is lined up with 3 divisions of the graticule at 0.9 of a millimeter. That means each graticule unit is worth 0.3 millimeters. Next, we want to remove the micrometer and add our specimen slide, making sure we don't touch the focusing wheel and take our magnification out of focus. We then line up the graticule with what we're trying to measure and use the scale, which we've already calibrated, to measure an object, a tissue, or a cell size on that new specimen slide. What I've done here for you is just put an image up of what that would look like. We keep our graticule as that stays on the eyepiece, and then we have plant cells here which we could now measure with our scale. If we said that the scale had 4 divisions for each plant cell from end to end, roughly, then that would mean that this plant cell is 1.2 millimeters in diameter or width. Now, if you're unsure exactly what I was talking about whilst those 2 things were happening, or you wanted to make some notes, here are the steps as follows. I've already said these whilst the video is going on, so I'll just show you the slides and allow you the opportunity to make notes for instructions if you want to. This is step 1, which is calibrating, and then there is step 2, which is when you measure using your graticule. I've given you an example there as well to make sure that you can work out the calculation yourself. Hopefully that helps, guys. And once again, with all of these skills that are practical, the best way for you to remember this is to have a go as many times as you can before the real exam comes around. This is a good opportunity to stop, as we've now finished on our calculation side of things. And if you are following along with my course, guys, complete task 1.7, which is to calculate magnification using a micrometer. It is also very, very helpful if you are studying to have a go at this on a real microscope, despite not needing to do this practically very often in an exam. The last skill is probably the most important because it comes up multiple times in practical paper 3, and that is drawing. It can be a cell drawing or it can be a plan drawing, and I'll show you what those are in a second. But students get this wrong most often because they don't follow simple biology drawing rules. I've listed those rules out for you on the right hand side, but I'm going to summarize quite simply now for you. Use a pencil to draw only what you can see, not what you think should be there or what you might be able to see. And when you're drawing using your pencil, not a pen, make sure to draw with straight, clear, continuous lines that don't overlap each other and that stay continuous on the paper. No dots, no shading, no light and dark. It sounds very simple, but it's easy to get wrong. Let's take a look at 2 examples of that. The first is a cell drawing. Draw and label the plant cell walls of the following image. Now I'm going to disappear again so that you can focus on the video, and hopefully you'll see exactly what you should do. Hopefully what you're seeing is solid lines, no shading and no drawing with artistic skill. I've only drawn what I've been asked to draw, which are the cell walls, and then at the end I add a single ruled line to add my label for the cell wall. This is one of the most common drawings that you'll be expected to do. Now you've seen how to draw cell drawings, let's just very quickly add one rule if you're asked to draw a plan drawing. Plan drawings are much larger in scale because you're drawing whole tissues or layers of a tissue that do not include cells. I added this slide specifically to teach my students, as well as you, not to draw cells. You only draw the outlines and labeled tissues. Please remember that, and let's take a look at exactly what I mean. And here is your example of a plan drawing. We are asked to make a plan drawing of the root vascular bundle, which is called the pericycle. You may not have seen something like this before, but don't worry, you'll learn it in the plants topic later in the year. Effectively, it's the xylem and phloem which is found in the root. I'm going to disappear once again and make sure you watch the video clearly whilst I talk to you about what I'm doing. So I start my plan drawing of the vascular area in the root by drawing two rings. The reason I've drawn two is because I can see a set of cells in the plan that are quite clearly surrounding the vascular tissue, so I need to show their cells. Once I've done that, I need to differentiate between those two types of cells that I can see within that ring. One's red and one's blue or purple. This is actually the xylem and phloem. I decided to add those two labels to my diagram to help clarity. And last but not least, I would like to make a point of saying I didn't draw any cells on my plan drawing, and neither should you. And once you've watched those videos, now would be a very good time to take another break and complete task 1.8 if you're following along with the course. Draw both cell and plan drawings from microscopy images. Again, with all of these skills, practice makes perfect. The more you have a go, the better you'll be. And it's very easy to practice this by looking at cell and tissue images on the internet and then comparing to real drawings from scientists. I've given you a few in my worksheet. Okay, once again, we've come towards the end of the video. We've finished looking at our skills, and so I'm going to give you some tips for the exam. These are my set of tips for your practical paper 3 microscope skills. I won't read them off because we're going to go through them one at a time now. So our first tip is using a scale bar for magnification calculations. It's an easy go-to tip so that you don't have to measure across a kind of unknown or hard length on a cell, and it also gives you units to work with. What I mean by that is you'll always be using the magnification equation with an image, but quite often, if you're smart, you can look underneath that image and there's a little scale bar there. It will have units and values on it. When you're working on by magnification, you can just use the scale bar instead of measuring things across the cell. That means that you're improving your precision and accuracy because you've got a known value already and you're measuring on a straight line instead of across 2 points, which could give you a bit of deviation. Nice and easy tip, scale bar, measure the bar instead. My next tip isn't so much of a tip, it's more of a revision method, and that is recognizing different organelles or parts of images from microscope images that you may be asked to draw or label, and when you're recognizing them. Find some free websites. One recommendation from me would be Wikipedia or Wikimedia, and take a look at cell diagrams and micrographs and scientists' interpretation of those, because then you've got something to work with. This is a skill where practice makes perfect, so practice. The most important tip that we have here on our tips section is xylem cell drawings. The reason why it's the most important tip is because it almost always comes up. An example of this would be draw 4 xylem cells, cells should be adjacent. So we know that we're not making a plan drawing and we have to draw the cells that we can see. Xylem cells are easy to draw because they have no contents. It is only cell wall. And once again, I'm going to show you a video. Again, here we have the pericycle of the root, and we're going to draw 4 adjacent xylem cells, and I would pick out a section like this. I'm going to disappear again to let you watch the video and show you side by side exactly how I would draw that. I'm not going to repeat what I've said before with regards to the drawing skill, but one thing that is worth highlighting here is the fact that I have drawn 2 cell walls next to each other. It's important that there are 3 distinct lines in between each lumen of a xylem, because this is a marking point in the exam. I've highlighted that for you on this diagram here. Micrometer questions can be particularly tricky, mostly because you're trying to draw information from the question, from images, and it's usually part of a larger question series. That means there's multiple steps. What you need to remember on these question series is, if you're asked to calculate magnification using a micrometer, then they must give you some kind of information or image to calibrate with. Don't overcomplicate the question and follow the steps from the instructions that we learned earlier. It's my best tip here. It is quite often very, very confusing, and so do have a go at some practice questions if you get a chance. Moving between units, as well as giving the units in your answer, is the most common way that students lose marks when using the magnification equation triangle. A boring, repetitive statement for me, but check the units you're given, check the units you're asked for, convert when needed, and make sure that the unit value, if you're giving image or actual, is in your answer line. The last tip that I have for you is actually more of a question that often comes up. It comes up at the end of the microscope section of the paper, and they ask you to compare two things. More often than not, this is a large tissue diagram of stems or roots or leaves in a monocot versus a dicot plant. Those are two different types of plant. When comparing, you need to make sure you use clear, measurable terms and that they're on the same scale. What do I mean by that? If you wanted to have a go, you could compare the left-hand monocot to the right-hand dicot stem cross-section, and you would start each of your comparative sentences with the following words. I've put these all up for you so that you can make a note, but you can see how they are all clarified comparisons, and you need to make sure you start each of your statements with those comparative terms. Hopefully that will give you a clear answer, and then you can get all of the marks in those big comparison questions at the end, guys. So that brings us to the end of all of the microscope section and learning. You should now be able to use this microscope, set it up, magnify, and use the magnification equation, as well as using a micrometer for that. And you should be able to interpret your diagrams and draw them with an appropriate scale. Well done for getting through the video, guys, and thank you very much for watching. Credit where credit's due. All of my images were either made by myself on AI, or they were from free licensed, free to use websites, which were Wikimedia, Pixabay, and FreePic. So thank you to those sites for the great images. I just wanted to take this opportunity to say thank you once more, guys, and for putting up with me jumping around the screen as a silhouette this time instead of a circle. That was from one of my students who said they would prefer me to be a floating head. Hopefully it's okay for you. And that should bring you now to the end of your revision and study for Unit 1, Cell Structure. The next video on biomolecules will be coming up within the next couple of weeks. If you do need any more help or exam materials, you can always visit my website, and I've left that up here for you now. Good luck with your study, guys, and thank you.